Welcome everyone to the inaugural Kolokotrones Symposium for the Childhood Glaucoma Research Network. I am Peter Chang. I am at Bascom Palmer Eye Institute and will be the informal host and moderator today. And we're very fortunate to have really two towering figures in ophthalmic genetics who will be speaking with us today. Just a brief word, our heartfelt thanks to the Kolokotrones family for supporting this lecture series, and we're hoping to make this a regular event. The goal is to educate people who take care of children with glaucoma and anterior segment diseases worldwide, so we can fulfill our mission of providing world-class care everywhere. And a quick word about the format. We will start with a lecture by Dr. Terri L, followed by a second lecture by Dr. Janey Wiggs. And the last third of the symposium will be devoted solely to questions and answers. So during the lectures, if you have any questions, don't be shy. Start typing them out onto the Q&A button. If you look at your Zoom screen at the bottom, use the Q&A button to type your questions. And the remaining panelists, Drs. Grajewski and Dr. Elena Bitrian, will sort them out and present them at the last third of the lecture. And just one other word, at the end of our lecture at noon Eastern Daylight Time, if you couldn't get enough of this and want to chit chat with colleagues worldwide, we have a separate Zoom link that will take you over to a virtual coffee break where we can chat, reminisce, commiserate, and have group therapy with each other. Okay? So with that, we're gonna start our first lecture. Let me take a few moments to introduce Dr. Terri L. Dr. Young is currently the chairman of the Department of Ophthalmology at the University of Wisconsin-Madison, and she's the Peter Duerr Endowed Professor of Ophthalmology. And one of the problems I have when I'm trying to introduce the two speakers today is that both of their CVs are longer than anything I've ever written forever, and it's really hard to pick out the distinguishing factors. But Dr. Young received her training, medical training at Harvard, Illinois Eye and Ear, and a fellowship at the Children's Hospital of Philadelphia. And as a researcher, she has been granted the R1 award in multiple decades. She will start off by giving us some refresher on ophthalmic genetics. So I'll hand the podium over to Dr. Young.
Unmute, sorry. Can you hear me now? Very good. Thank you so much for the kind introduction. I am truly honored and grateful to be asked to present, and I'd like to thank you and also Dr. Grajewski for the invitation. I'm also honored to partner with Dr. Janey Wiggs, who is a medical school classmate and glaucoma genetics colleague. And also to the Kolokotrones family for this, this lecture series. Can you get to the first slide, please? I think you're going to handle it from that end because I'm literally on my phone. Well, I just wanted to start by saying that I do have one disclosure, and that is that I'm a consultant for AirPO Pharmaceuticals. I don't know if you can pick up or put that slide on the screen, please. There we go. Okay, super. Thank you so much. Next slide, please. So, yeah, I've been at this gene hunting business. I'm a pediatric ophthalmologist, and I've been at this gene hunting business since the 1990s. So I'm an old lady at this in many respects. And I just wanted to say that, you know, back in the day when we were using different kinds of technologies, I'm really just pleased that because of the technological and statistical advances that we have now, it's really allowed us to realize, at least me, my ambitions to find genetic causality for devastating ocular disorders and develop directed biologically based therapies that are curative for those disorders. And I think that we're really on the cusp for that in congenital glaucoma in particular, again, because of the advances in technologies, which I will go through with you. Next slide, please. So there are 30,000 genes in human DNA. So there are about 3 billion base pairs that contain all the information necessary to make a human being. You know, that's a pretty lofty goal, if you will, but the hunt actually started in the 1990s with the Human Genome Project. And there were declarative statements, if you will, around the 2000s, 2003, that we had actually sequenced the whole human genome, and therefore could then start to, you know, kind of do deeper dives into the particular genes within that are that are encoding proteins that are then, you know, making different types of tissues to allow us to, you know, figure out what's happening biologically with genetic disorders. Next slide, please. In some ways, I see searching for disease genes as pretty much as like using your GPS. It's a car ride, if you will. And you kind of start off with a whole genome, those 30,000 plus or minus genes in human DNA, and you hone in on different locations, and those locations get more and more, you know, sort of specific, if you will, you know, and so you get down to a location, which could be a city or a particular chromosome. And as you know, there are 23 pairs of chromosomes. 22 pairs are marked as autosomes from 1 to 22. And that last pair are sex chromosomes. And then you get down to a band on that chromosome, because you can actually visualize different types of bands by staining. And then ultimately to a gene. The bands and the gene, the chromosomes there's a P arm and a Q arm. The P arm's the smaller, the Q arm's the larger. And then to the gene, which is where you actually get to different base pairs. So very specific changes in base pairs or a set of base pairs. Next slide, please. You've got to go through this one because there are different ones. And what's helped us is what you can see has happened, especially in the 2000s, is that both the type of process, which started off with mapping and linkage and homozygosity mapping and restriction polymorphic enzymes, or different kinds of primer pairs that had known locations on specific locations on chromosomes, to genome-wide association studies, to deep sequencing technologies. In addition to, you know, the type of technologies, the cost actually became actually affordable. And now what used to cost hundreds of thousands of dollars is now costing hundreds of dollars, if you will, to get the same information or even more, especially with our deep sequencing technologies. Next slide, please. So we got to go through some terms and some concepts here. Causative genes are genes that are directly leading to that disorder. There's a recognizable inheritance pattern, and we'll go through that, but I know you're familiar with autosomal dominant or autosomal recessive or X-linked inheritance or mitochondrial inheritance, where it might be passed down from the mother through her egg. Generally, there's one gene per family. We call these private mutations. These are less common diseases, but devastating diseases, things like cystic fibrosis or the muscular dystrophies. In the ocular world, it might be albinism or retinal dystrophies or corneal dystrophies. And then there are susceptibility genes, which confer an increased risk, but do not necessarily directly cause a disorder. There might not be a clear inheritance pattern. It involves many genes or genes and environment, and they're common in the population. And generally, these kinds of genes are discovered through, larger data sets of hundreds of thousands of individuals who have been well characterized, if you will, and in terms of their clinical features. But these are the kinds of susceptibility genes that, in some senses, line our hospital beds in our clinics: cancer, heart disease, dementia, myopia, progressive open-angle glaucoma, age-related macular degeneration. For the causative genes, we generally look for those in families, and the larger the family, the better, because we can do something called co-segregation analysis to see if that gene actually only affect the affected individuals in that family. But again, it's usually a private mutation, one gene per family. And for susceptibility genes, as I said, we usually need very large data sets, which I know Janey will go into, in the hundreds of thousands. And now we're doing meta-analyses and larger analyses, combining data sets of different population studies and different ethnicities. Next slide, please. There are different disease types as well. Next slide, please. Mendelian disorders are usually due to a defect in a single gene. And the premise is based on the color of Mendel's flowers that cause peas. In this example, autosomal dominant example here, the parent had yellow flowers, if you will. So, and that's dominant, so the YYs are in caps. The second parent had green flowers, which were recessive, so the YYs are in smaller case. And the offspring in this situation are all yellow. The offspring could have a big Y from the first parent and a small Y from the second parent, but the flower would be yellow just because the dominant larger Y takes precedence, if you will. Examples of this might be Huntington's disease, certain retinal and corneal dystrophies, various types of ocular albinism. Another example would be if both parents had the two small Ys, or maybe one parent had a big Y and a small Y, and the second parent had the two small Ys, then there is a statistical chance that the offspring would have a green flowers instead, because they would have the chance of having two small Ys, just like one of the parents. Complex diseases is kind of like a hand waving, if you will, in genetics. Risks is influenced by many genes. There are environmental factors that are thrown in there and other interactions that we may or may not understand. And I call this a sort of a Mendel's pea soup sort of situation. Again, in the ocular world, age-related macular degeneration, myopia, progressive open-angle glaucoma would fit in that category. Next slide, please. The other thing that we talk about are phenotypes. Those are the clinical characteristics, the biochemical characteristics, the physiologic characteristics of an individual that is determined by his or her genotype and the environment in which it is expressed. Here's an example of cystic fibrosis. They've got lung disease, clogged lungs because of mucus clogging and other issues along with that. Thank you. And then genotype would be the genetic constitution of that individual at a given locus or location. and that would be the two expressed mutations, if you will, for this autosomal recessive disorder for both chromosomes that are provided, one by the mother, one by the father. Next slide, please. A locus, as I said, is the actual position on the chromosome that is occupied by the gene on that chromosome. So it's and then the allele is one of the alternative versions of a gene that may occupy that at that given locus. And so we're given one allele by our mom, one by our dad. So again, those are important terminologies. Next slide, please. So there's heterozygosity, there's homozygosity, and there's hemizygosity. Perhaps hemizygosity is the most recognizable thing to understand, especially for the sex chromosomes. You know, if you are a male, you have one X and you've got one Y. So you're going to only have one representative chromosome or chromosome segment. And that would be a hemizygosity state. Heterozygosity means that at an individual or genotype with two different alleles at a given locus. So your mom gave you that small y, your dad gave you that large y. So you would be heterozygous. You would have heterozygosity at that particular location or locus because you've got these two different alleles provided by your parents. Homozygosity would be an individual or genotype with two identical alleles at a given locus. So that would be maybe your mom gave you that small y and your dad gave you that small y as well. So two autosomal recessive traits gives you an autosomal recessive phenotype or expression. Next slide, please. The other thing I wanted to explain here was when we do look at a gene, there are different, there's a structure and different regulatory elements for that gene. Exons are the coding sites of a gene. They're in blue there, so the cat, 8, and then there's hat. The alternative splicing is purple because the was probably an exon in this situation that was spliced out, and that happens with genes as well. We have introns, which are allegedly non-coding sites within that separate exons after there's a promoter site at the beginning and a poly A tail signal at the end that defines the entire gene. We're finding that there are intronic messages, if you will, that also influence the coding of that gene. And those are expressed here in black. And then there are these untranslated regions, ones at the 5' end or the beginning of the gene, untranslated region or UTR, and the others at the 3' region of the gene, 3' UTR. Next slide, please. The other issue is that there are gene to phenotype correlations. So I'm showing you all these other things. It's not just let's find this gene on this chromosome. There are many nuances to going down this car ride, if you will, to find this gene. In some ways, it's kind of like dodging cars because there are many things to think about in terms of whether or not this gene is causal. For some heritable eye disorders, for instance, that are shown here, like Duerr honeycomb retinal dystrophy, there's only one gene, mutations of which cause this particular retinal dystrophy. Another example for one to many would be the big H3 gene, mutations of which cause different kinds of corneal dystrophies, granular corneal dystrophy, avelino-dystrophy, and then retinitis pigmentosa is a large basket of many to one, where there are many genes that cause different types of retinitis pigmentosa that visually and clinically to us show the same changes, and we're calling it this, you're putting it in this one big basket called retinitis pigmentosa, even though there are multiple gene mutations that cause this disorder. And then there are many to many in disorders like age-related macular degeneration or myopia that cause the same disorder, if you will, that we're characterizing it as or categorizing it as, but many genes cause the problem. Next slide, please. We do different, keep pushing there because the pictures will come up. We perform different kinds of sequencing. There are two methods. One is the classical Sanger sequencing, which requires a single-stranded DNA template, a DNA primer, which is a segment of DNA that is that defines that gene and is at a known location on a known chromosome. A DNA polymerase, normal nucleotides and modified nucleotides that terminate the DNA strand elongation and are fluorescent. And when they bind, they terminate the DNA strand extension and result in a DNA fragment of varying length. And we use fluorescence to detect during the process of the chain elongation, a DNA sequence is elucidated. In next generation sequencing, the DNA is randomly sheared between 150 to 200 base pairs, and adapters are ligated to both ends of those fractions. There are these RNA capture probes are hybridized to prepare the DNA fractions. And then this hybrid selected enriched output library is amplified by polymerase chain reaction. And then subsequently, the reads are aligned to reference the genome and analyzed for single nucleotide variants. So these are longer reads. And it's not one base pair at a time like in Sanger sequencing. When we find something by next generation sequencing, and we do this by whole exome sequencing or whole genome sequencing, looking at the exomes and the intronic spaces, we go back to Sanger sequencing to verify that in families and in individuals, and that's called co-segregation analysis. Next slide, please. Some of the changes in base pairs lead to different amino acids. As you know, 3 base pairs code or encode a particular amino acid. So you could start normally with the UUU, but if you change that 1 base pair, last one to a gene, then the phenylalanine becomes a leucine, and that could have consequences for the protein that it's encoding, from everything from its folding to its hydrophilic or hydrophobic nature to its ability to couple with other proteins, especially like if it's a receptor or a ligand. Nonsense changes are a change in a base that leads to a premature stop codon. So a UUA encodes a leucine, a UAA encodes a stop codon, so that the gene no longer continues to be translated. Next slide, please. The other things we have are in-frame deletions and insertions, different types of amino changes. Next slide, please. We also have these chromosomal rearrangements. The most familiar one is Down syndrome, which generally is a third chromosome 21 or a third portion of chromosome 21. You've got examples here of deletions or inversions as examples, and these could be dominant or recessive, autosomal or X-linked disorders. Next slide, please. Now, SNP, or single nucleotide polymorphism, is a little different. Again, these are larger population studies. These are changes usually in a base pair that is different in a population. And we see that it confers risk because it seems to be weighted more towards a certain disorder. And so we do genome-wide association studies to assess these. Next slide, please. I'm being told I need to zoom through. Next slide, please. These SNPs are usually carried down through different generations or historically through, and you can see through, if you will, kind of like the diaspora of how people migrated throughout the world, how these SNPs are carried down through historically, if you will. Next slide, please. The approach, you have to use thousands of individuals across and use very high density SNP chip arrays. The outcome is a Manhattan plot. You look at the top, and each of those colored bands are a different autosomal chromosome with the X chromosome at the end, and they form plots with different statistical analyses showing generally with a p-value of 10 to the minus 8, some significance of different SNPs on a particular gene or outside of a gene or near a gene in an intronic region that are more likely to be associated, if you will, with that particular disorder. And that's how we find causality or susceptibility, if you will, for different SNPs. Next slide, please. So for GWAS, you're going to find genetic changes, generally a small effect, that are ancient, relatively common. At the other spectrum, for deep sequencing or Sanger sequencing, you're going to find more private mutations with a larger effect phenotypically. And these generally are newer genes or even de novo, meaning that it might be manifest for the first time in that individual. Next slide, please. So what happens once we identify candidate genes? Determine gene functions, evaluate gene variations. Our hope obviously is it's an improved clinic management. We are hopeful for more accurate and earlier diagnosis, more accurate prognosis, and obviously to develop better treatments and right drug to the right patient, which is our hope for precision medicine, get the right pharmacogenetics to the right patient. Next slide, please. And then there are therapies or interventions that we can introduce if we know what this person might be in for, if you will, because of their genetics. Possible lifestyle changes in terms of changing their diet, different educational program, maybe doing a surgery, chemotherapy for treatment or prevention, and certainly genetic counseling, genetic counseling, genetic counseling. Next slide, please. And as I said, pharmacogenetics is this gene that, this dream that is happening right now, that we can develop a sort of a genetic profile, which in turn is a biological profile for an individual that says that they're more likely to respond to this, this formulation of treatments, pharmacologic treatments or other treatments for disease, especially in cancer, than this profile, given this profile with treatments. And so we can give them more directed therapies to really attack the disorder, especially in cancer, in a more thoughtful way. Next slide, please. So I've gone through multiple steps: interpretation, mutation versus polymorphism, does the result match, the phenotype, the pedigree, segregation analysis, prognosis, what else to look for treatment. Next slide, please. Which test to order? It's critically important of the phenotype. If it's a clear phenotype like Down syndrome, targeted tests like a karyotype might be all that's needed. If there's genetic heterogeneity, like in retinitis pigmentosa, you might want to do a panel of specific genes tested as a group. If there's more than one system, like if it's a eye disorder with a kidney disorder with a cardiac disorder, you might want to do a microarray. If you have no clues or matches or targeted tests and they're all negative, consider whole exome or whole genome sequencing. Next slide, please. And genetic testing has been happening for some time. Newborn screening has been happening for decades. Panel of recommended 34 conditions in general, but these are state-based and state-prescribed programs Generally, these tests are for metabolic disorders, and parental informed consent is not typically required in these situations. Next slide, please. But there are also different kinds of thought processes in terms of the types of genetic testing we do throughout the life cycle. Depending on if it's early childhood, as I mentioned, karyotyping, maybe gene panels, microarrays, or exome sequencing, depending on the disorder and how recognizable the phenotype is. For adolescents or young adults, you might go to gene panels, microarrays, or even go straight to exome sequencing. There's carrier testing, and there's predictive and predispositional testing as well. Next slide, please. And just as a wrap up, you know, these gene testings create new opportunities, but also present new challenges. The interpretation of the variants could be difficult. Not necessarily every gene test gives you the variant or the enough of the information that you might necessarily need. Sometimes these variants are of uncertain significance. Sometimes you might identify variants that are unrelated to the indication for testing, and what do you do about that? What if you, for instance, get mutations for breast cancer, when the BRCA1 or BRCA2 gene mutations? Do you tell the patient about that as well? What do you throw in the bucket? And then a big one is may reveal misattributed parentage or even consanguinity or incest. And how far do you go with informing the family and the patient of that, and what kind of consequences might lead there too? Next slide, please. The other negative consequences or risks are general. The burden of knowledge, it can create anxiety or guilt, especially if it's a disorder that doesn't manifest until later in life. It could change the relationship of that patient with other family members. They may not provide the same resources or educational circumstance to that particular child, for instance. It may affect plans to have children or create moral dilemmas around that. And then there's a concern for potential impact on health and life insurance. Next slide, please. And then there are big challenges, especially with pediatric genetic testing. Whole genome testing in children as a first-tier test. You're going to get more information than you perhaps need in that situation. There's secondary findings and when to report them. Do you want to necessarily do carrier testing in adolescents, per se, and then give them that information and that burden? People are doing direct-to-consumer testing on children and making lifestyle changes, educational changes based on that kind of testing without the intervening geneticist, genetic counselor, primary care doctor providing an interpretation of those kinds of testing results. Folks are asking for genetic testing before they'll adopt a child too. And what does that mean? And consanguinity and paternity, how and when to report that. It can be very, very difficult. So in conclusion, next slide, please. We cover the basics of genetics. We cover the basics of gene hunting. We cover the current recommendations and implications of genetic testing throughout the life cycle, from newborns to young adults. And I wanted to just do this as a primer 101, and thank you again for your kind attention. Next slide are my references, and thank you.
That's wonderful. Thank you very much, Dr. Young. I think that was a masterful lecture, and anybody who can distill the genetics basics down in 20+ minutes and make me understand it really truly has honed their craft. So, we're very lucky today. We get to have 2 entrees, so that was the first one. And now let's move on to Dr. Janey Wiggs, who really in my mind requires no introduction, but here we go. Dr. Wiggs is the Paul Chandler Professor of Ophthalmology and the Vice Chair of Clinical Research at the Massachusetts Eye and Ear Infirmary, Harvard Medical School. And she received her bachelor's and doctorate training in biochemistry from UC Berkeley, followed by her medical training at Harvard. And I believe residency and fellowship are both at Harvard. And as if that was not enough, Dr. Wiggs went on and completed 2 genetics fellowships in the Boston area. And similar to Dr. Young, she has been a very successful R01 grantee for multiple decades. She is the inaugural chair of the genetics group of ARVO. And she's one of the very few ophthalmologists, researcher who's dual boarded in both ophthalmology and medical genetics. So with great pleasure, let me hand the podium over to Dr. Wiggs.
Thank you. Thank you so much, Peter. And thank you, Terri, for really setting a nice background for my talk. If I can get this to start. Okay. So I'm really going to talk about the nuts and bolts of genetic testing, and specifically genetic testing for childhood glaucomas. It will be kind of a practical primer that follows the instructional primer that Terri just gave. So my disclosures, I have grant support from NIH. A grant from the March of Dimes has supported some of our work that we've done for juvenile and congenital glaucoma, and I also receive grant money from the RPO company, and I'm a consultant for a number of other companies. I also want to disclose that I am the director of our CLIA certified genetic testing laboratory at Mount Sinai, which I don't think biases me about genetic testing for childhood glaucoma. I can just say that, in fact, I've been a believer in genetic testing for all inherited diseases throughout my career, and am especially passionate about genetic testing for glaucoma. So the overall goals of glaucoma genetic research, and this is, you can really apply this to any disease, but in particular for glaucoma genetics, is to identify genes that cause or contribute to glaucoma. That is the goal, using some of the methods that Terri outlined. And the outcome of that is, first of all, we can define the underlying molecular events that are actually responsible for disease. So by knowing what a gene, what a gene's protein product normally does, and then seeing what it doesn't do or abnormally does in disease, we can begin to understand the molecular mechanism of disease, which can suggest then novel therapies that actually target that particular or specific molecular pathophysiology. And in addition to that, and I think really importantly in addition to that, we can use genes that are discovered as causative for glaucoma for genetic testing. And that is going to be the focus of what I'll talk about today. So why would you want to do genetic testing for childhood glaucomas? Well, first of all, the genetic test results can really help inform genetic counseling and risk assessment. And this is because the early onset glaucoma genes can produce clinically similar phenotypes, but that have different inheritance patterns, and therefore have different risks to other family members. So, for example, in these photographs I've shown here, these are 4 patients with early onset glaucoma, and 3 of these 4 have mutations in genes that lead to autosomal dominant inheritance, FOXY1, PIDX2, and 2 patients here have FOXY1. So what this means is that the other family members or children of these individuals have a 50% chance of developing the same disease. While one of these patients, the patient who has mutations in CYP1B1, that's inherited as an autosomal recessive trait. And so therefore, the offspring of this person or other family members has a reduced risk of disease as low as lower than 25%. So let me give you a couple of examples of how we actually use this in the clinic. Here are 2 patients that I've seen in the glaucoma clinic at Mount Sinai. They were both diagnosed with early onset glaucoma early in life. The patient on the left was diagnosed at birth with intraocular pressures of over 30. Patient on the left who had a cloudy cornea at 1 year with also elevated intraocular pressures. Both patients came without any known family history of glaucoma in siblings or parents. So we did genetic testing on both of them and found very different results. For the patient diagnosed at birth, the genetic test revealed a mutation in the gene FOXC1. As I mentioned, this is inherited as a dominant trait. It turns out that the mother actually had this same mutation, but this is a gene that's notorious for variable expressivity or incomplete penetrance. And she actually wasn't aware of any problems. She didn't have this severe glaucoma. And we observed this in these families where the child has the mutation in severe disease. So here, we recommended testing other family members and would advise the family that, other family members have about a 50% chance of having this mutation. The other patient, turns out, had mutations in CYP1B1. That's recessive inheritance. Both parents are carriers. One of the siblings was also a carrier, but no one else in the family had 2 mutations which are required for this disease. And again, then, the risk assessment and counseling that you would offer to family members is very different for this recessive disease than for the dominant disease. So another reason you would want to do genetic testing for childhood glaucomas is to be able to target within a family, to be able to target surveillance and treatment to people who actually have the mutations. Oftentimes what happens with these families or individuals is that there's one person in the family who has disease, and they bring the whole family in every 3 to 4 months for an exam because you don't know if the other kids are going to develop the disease or not. By genetic testing, you can identify everyone in the family who has the mutation and target your surveillance and your testing and your treatment to the patients who actually have the mutations. So here, this is a simple pedigree just to illustrate this point. This father, the older individual, presented with decreased vision at age 40. When we did his visual field test, we found very significant changes and his pressure was slightly elevated. So because his disease was so advanced at this, relatively early age for glaucoma, we did genetic testing and found, in fact, that he had a myocilin mutation. So then we tested the rest of the family, and we found that his son also carried the myocilin mutation, but only had modestly elevated pressures and didn't have yet any visual field changes. But what we can do now, knowing that he has this mutation that causes disease, is that we can follow him very carefully and initiate treatment at the very earliest stages of disease. Importantly also, we noted that his daughter did not have a mutation, and therefore his daughter does not need to have increased surveillance. Her risk of disease is no greater than that of the general population. Another reason you would want to do genetic testing for childhood glaucomas is because there are likely to be gene-based therapies coming into play in the next 5, maybe years, and that it's important to identify patients who have mutations in genes that would benefit from these novel gene-based therapies. And so I offer here one example, which is myocilin. I think the one that's most likely to come into clinical practice the earliest. Myocilin is, of course, a gene that causes juvenile glaucoma. Mutations cause the protein to aggregate in endoplasmic reticulum in the cell, causing the cell to die and therefore causing elevated intraocular pressure. In a proof of concept preclinical study, Val Sheffield and his colleagues at Iowa have shown that gene editing using CRISPR-Cas9 removes the myocilin mutation, or the mutant myocilin, and lowers intraocular pressure in the mutant mouse. So this is an exciting advance and suggests that this kind of therapy could also potentially be available for humans in the near future. So what do we do when we do genetic testing for early onset glaucoma? Well, we currently test for all the genes that we know can cause childhood forms of glaucoma, and they're all listed here on this slide. So a relatively small number of genes if you're thinking about things like inherited retinal degenerations where there's over 250 individual genes that can cause disease. There are 4 genes that can cause primary congenital glaucoma thanks to the great work that Terri and her colleagues have done to identify TAG or Tie2 and angiopoietin-1 most recently as causes of congenital glaucoma. There are 4 genes that can cause anterior segment dysgenesis type forms of childhood glaucoma. And keep in mind that these do not always look like abnormal anterior segments. Sometimes, especially FOXY1, they can look really quite normal. Myoclonus, as I mentioned, cause juvenile open-angle glaucoma. And then there are 2 genes that cause familial types of normal tension glaucoma. So you'll find that, in fact, in some of the gene testing panels or a number of other genes that are listed as also childhood glaucoma genes, and the evidence, for genes that we should look at is very important to know that, in fact, a gene is actually causing a disease. This is an initiative that has been undertaken for all inherited diseases by the program ClinGen, which stands for Clinical Genome Resource. It's a trans NIH supported program supported by the National Human Genome Research Institute. And the goal here is to build a resource that defines clinically relevant genes and variants to be used for precision medicine and research with the overriding goal of asking, is this gene associated with a disease? And is this variant in the gene causative? And then what is the clinical utility and relevance of that? So just in the past year, we have formed an ocular domain working group within ClinGen, which has been a great opportunity to look carefully at genes in the literature, for their construct validity, for actually contributing to childhood glaucomas. And so we're currently curating the childhood glaucoma genes, and this will be, of course, an ongoing process. As more genes are discovered that cause childhood glaucomas, we'll have the opportunity to put them through this ClinGen process to determine, their actual validity in terms of causing disease. So one of the current limitations, as you can imagine, with only 10 or 11 genes that we know that we're confident can cause childhood glaucoma, is that the diagnostic yield for these known genes is relatively low. Even if we employ things like whole exome sequencing, which can identify some novel genes, overall, we only detect pathogenic alleles or mutations in about 20% of cases. So we really need more research in this area. We need more effort to find, additional genes. I feel quite fortunate that we were just awarded a grant from the National Eye Institute, or NIH, to do whole genome sequencing for a number of these early onset families that we've studied for years, to look for, a very comprehensive set of genetic variation that could potentially influence the development of these early onset diseases. So how do we actually do this genetic testing? Well, here is just a cartoon that goes over, kind of the logistics of doing genetic testing. It starts with the patient who's referred for genetic testing. That can be from, an ophthalmologist, it can be from a geneticist, it can be from a primary care physician. We get referrals from many different types of healthcare professionals. Next is to have a biological sample. Like, we need to have material to purify DNA from, and that's either blood. Actually, everyone always prefers blood, but with children, sometimes that can be difficult. So sometimes saliva samples, cheek swabs can also be used for this kind of testing approach. We need to select the appropriate test. Terri talked a little bit about this. There are many different ways to do testing. Sometimes you want to do a single gene test if you're absolutely confident that the phenotype is this one gene. But most of the time, because of this phenomenon where a single gene can have a number of different phenotypes, or there can be a number of different phenotypes related to a small set of genes, it's actually quite helpful to do a panel test where you test multiple genes at once, or even do whole exome or whole genome sequencing as the initial test. So all of these tests require bioinformatic analyses. The more complex the test, the more complex the bioinformatic approach is, because you need to take all of these variants and all of these DNA changes that you get from a single test and kind of narrow focus down on the ones that are most likely to cause disease. And that involves a number of bioinformatic steps and to remove variants that for various features are not likely to cause disease. And the test report is then written by a genetic analyst or a genetic counselor. The lab director reviews and signs the report. The report is then sent back to the healthcare provider with recommendations for follow-up. So we generally, for childhood glaucoma genes, and actually for all the eye disorders that we test for, we generally do a panel test. And the reason for this is just because of this relationship between, this complex relationship between genes and phenotypes. So our panel test includes all of the early onset glaucoma genes, and we also test for some copy number variations, which are not typically identified using approaches such as Sanger sequencing and things like that. It's helpful, to have this panel test because we also sometimes identify mutations in genes that are not suspected by clinical exam. For example, somebody who has a FOXY1 mutation but has a perfectly normal looking anterior segment without any signs of anterior segment dysgenesis. And actually, the panel test cost, because it's more efficient, is lower and faster than traditional sort of Sanger sequencing. So I think interpreting the report is something that is important to understand. I'm just going to really touch the tip of the iceberg here with this, but I think this can confuse people when they get the reports and they're like, what does all this mean? And basically there are several things that are critically reported. The first is whether or not it's a positive test. If it's a positive test, it means that a mutation likely to cause disease was detected. As I said, about 20% of the time for glaucoma, early onset glaucomas, we can do that, and that's a wonderful thing. A negative test means we didn't find anything. So nothing, and again, this sometimes is quite helpful, although it doesn't mean that the patient doesn't have a mutation in a gene that we haven't found yet. Unfortunately, variants of unknown significance or uncertain significance is something that we find quite frequently. So we'll find something that looks good, it's rare, it could have some pathogenesis to it, but it hasn't been validated in the literature with any kind of functional testing. It might be in a gene that might be related, but maybe not. And so, we can report this as, well, this is interesting, but we can't be confident that this is really causing disease. Terri mentioned about the incidental findings. This is a mutation that has clinical relevance, like a mutation in a breast cancer gene like BRCA1 or 2, that the patient should know about, but it's not really what they came to be tested for. And so that can be something of an ethical problem. ClinGen has published 59 variants that need to be reported if you encounter them. Usually you only find these if you're running a whole exome or a whole genome test. In our panel test, we only are looking at glaucoma genes or eye-related genes, and so we don't typically encounter these. And then the report may recommend that there's family follow-up, because that can really help confirm the causality of any mutations that are identified. So who do we test? Well, I have a low threshold for testing, as you could see from the pedigree I showed, but that's because studies have been published that suggest that patients who have disease glaucoma before the age of 40, or those even with advanced disease before the age of 50 are more likely to have mutations in these genes that we know about so far. We also recommend testing for patients who have a primary family member, like a mother or father or a child, who also have this same disease onset before age 40 or advanced disease before age 50. So where does the testing occur? Well, genetic testing needs to occur in CLIA certified laboratories. And this is the CLIA Act was approved in 1988. It's administered by the government. It's to make sure that certain standards are met for, for testing in terms of quality control and proficiency testing. We want to be sure that we offer safe and accurate and also accessible tests. There are a number of CLIA certified laboratories in the United States and throughout the world that offer genetic testing for eye diseases. I'm not going to list them all here. I list a link if you want to look them up. The one thing I would say is that I think some are, academic like ours and some are industry. And I think that the one thing that's really helpful is that it's helpful to establish a relationship with a laboratory, because then you become familiar with their tests. And so it makes it easier to know if something's an outlier or something is, actually significant when you get the test back. Our work is also, these genetic testing guidelines that I've suggested to you are also consistent with the more recent recommendations of the American Academy of Ophthalmology Task Force on Genetic Testing, and that's been published on the AAO website. So just to summarize then, this kind of nuts and bolts talk, why do you want to do it? You can inform genetic counseling and risk assessment, identify people who really need to be followed for disease and treat them early, and identify patients who could, in the future, benefit from specific gene-based tests. What do we test for? Current known genes that cause Mendelian forms of glaucoma. This is an ongoing list with efforts underway, like programs like ClinGen and new gene discovery. We'll have better, hopefully more complete lists and be able to offer more comprehensive testing. Panel testing for childhood glaucoma is actually the most useful, just because of this complex relationship between genotypes and phenotypes. Patients with disease onset before age 40 or advanced disease before age 50, that is age 55 is a typo, but I guess 55 is okay sometimes too. And then where in CLIA certified genetic testing laboratories is where the testing should occur. So, in this field, it's a nice field, very collaborative, and we really need to work together to identify additional genes. We only have 11, we need more. We need to understand the relationships between genes, mutations, and clinical outcomes. This is really an important part of, you find a gene, but you need to know how that gene impacts the clinical disease. And that's where things like ClinGen and ClinVar come in, but also just the great work that everybody does in their clinics to correlate, clinical features with specific genetic mutations. And then, increasingly, we're going to be identifying targets for gene-based therapy. I mentioned myosin, TEC and angiopoietin-1 are not too far behind, where there's a clear path there, potentially also for treatment, and potentially others as we find other genes. So I just want to acknowledge people in my lab who work with me on these early onset glaucoma projects, the Ocular Genomics Institute, where our CLIA lab is located, and in particular, Emily Place, who is our genetics counselor, who just is incredible and does amazing work. And then funding from NIH and March of Dimes. And also, I want to acknowledge and thank the Kolokotrones family, who has donated some money for glaucoma research to Mass Pioneer, which in fact I was able to use for some of the work that I've done on this in this area. So thank you very much.
Thank you, Dr. Wiggs, for what I thought was just a virtuosic summary of genetic testing in childhood glaucoma. And it's worthwhile to mention that you talked about the CRISPR-Cas9, and the developers of the system was just awarded the Nobel Prize in chemistry. So I think that's very good evidence that genetic testing is an integral part of what we need to do when we take care of these patients. And so, as I jokingly said early on in the introduction, that we run on Miami time here, and because of how wonderful the lectures have been, I speak for the group that we can extend this symposium by approximately 12 minutes. So I'm going to hand the microphones over to Drs. Elena Bitrian and Dr. Alana Grajewski, who will go over some of the questions that the audience have, and actually have received a couple via text also.
Hello. Yes, so we're going to answer some of the questions that have been posted during the amazing talks we just heard, and feel free to add more questions to the chat if needed. So one of the questions we received is if an eye doctor does not have access to a geneticist or genetic testing in the area, and the patient is not able to travel, how do we go forward to pursue genetic testing? Can these be sent? Can the samples be sitting? How is the shipping of the blood? What do we do? Where do we send it?
Yeah, so I'll take that. We receive samples actually from all over the world for genetic testing. Admittedly, a lot of samples come from clinics at Mount Pioneer, but the DNA samples or cheek swab samples can be sent through the mail, and actually the whole process can be arranged externally. There's no need to actually come into a clinic. I think that so that's for the testing itself. I think that, we also do genetic counseling, which, we can do virtually. And thanks to the wonders of Zoom and other things like that, it's actually one thing that really can be done using Zoom. But, I think it's really important to have, if they're, to have some local expertise as well, just in terms of like follow-up and so forth. But certainly it all can be done externally, virtually, and, and it's something that's done quite commonly.
One of the related questions that came through had to do with cost and what to do if insurance doesn't pay for this, and how do you know if insurance will pay for it? So how do we answer those questions for our patients?
Yeah, so that varies, of course, from laboratory to laboratory. I can tell you what we do. We for every patient, we look at their insurance and we know which insurance companies will pay and how much they will pay for the testing that we do. Early on, we worked really hard with the Massachusetts payers, to demonstrate medical need for these tests. And I think increasingly, insurance companies are understanding that there's a role for genetics in clinical care. And I think that going forward, this will become less and less of a problem. But so what we do, before the patient sends any kind of sample or anything like that, is that we counsel the patient on, what the insurance will pay, how much it's going to cost them, and, so that they know, upfront what the situation is. And I'll say also that the costs are, decreasing, and some of the companies in particular are offering, lower and lower costs for doing this kind of testing. And so I think there's, there actually is a, that kind of barrier, which was probably more substantial 5 or 10 years ago is increasingly becoming less and less.
I would also say too that you know, there are some states that are more liberal, if you will, about this than others.
Yeah.
You know, Wisconsin actually does cover some of those costs as well. It's just sort of, you know, kind of part of the state budget now, although that might be changing with COVID-19. But it does depend on the sometimes the relationship of the state hospital, if you will, with the government in terms of coverage of those types of testing. So it's worth probably working with the genetic counselor in your system, because they would know those different avenues.
Terri, this question's directed towards you. What are the biggest barriers to identifying more of the genes causing early onset glaucoma? And how can providers caring for pediatric glaucoma patients help to move this field forward?
Sure. Just like Janey, and then Janey, you know, we've collaborated too. We have collaborators from throughout the world who are, you know, they care for patients with congenital glaucoma, or they themselves are involved in research in their laboratory settings throughout the world. So the biggest barrier, you know, is just having enough volume, enough numbers, if you will. And I would say the other thing would be, you know, because of what we're finding is not just getting the DNA from the one affected individual, the proband. It's to actually expand these families. Janey showed you 2 examples of not just the proband, but the parents and the siblings, and actually genotyping them as well, so that we can have a better understanding of how the disease manifests within a family, and you know, make sure, especially with collaborating with other groups, that your lab isn't the only one finding a mutation in that new gene. But I would say, you know, it starts with practitioners who have patients with this disorder, understanding that they need to be, you know, part of the, you know, kind of like part of the foundational makeup of finding these genes, and identifying a researcher or a lab or a consortium, you know, that would happily partner with them in terms of getting the DNA, getting the phenotypic information for that family, and then having that that family undergo the process of generally exome sequencing or whole genome sequencing to get to the answer. So it really is you know, very important for these partnerships to happen. I know, you know, sometimes it's cumbersome for, you know, practitioners to get involved in all that. So it might mean that they have, you know, sort of set up a local circumstance where they partner, say, with a genetic counselor who can help them gather that information, either a counselor or sort of a clinical researcher, you know, administrative assistant, to get that information and collect the DNA from the family, and get the consents for, you know, genetic testing, and then, you know, send it on to the respective lab. But it's just really getting that partnership going.
Thank you.
And so as a whole aggregation, how do we see, we have the Childhood Glaucoma Research Network. Do we see a role for that to bring all the specialists interested in pediatric glaucoma?
Elena, you want to answer that question?
I'm actually not sure I understood the question. What was the question?
So we do have the Childhood Glaucoma Research Network that brings together different specialists on pediatric glaucoma around the world. Do you see any value on this association as well?
Absolutely. And you know, I've been attending those meetings from the onset. Absolutely. I think that, you know, and I've mentioned at these meetings that, you know, you have fabulous collaborators who are now doing a lot of work in the sort of standardizing the sort of the clinical characteristics of childhood glaucoma. We need to bring that forward with an actual DNA biobank, if you will, maybe even follow with tissue biobank for those that undergo surgery, so that we can take that to the research laboratory to to identify actual genetic changes in these patients that can be brought back to the patients and to the clinician, so that thought processes for directed therapies can happen, as well as all the other things, surveillance, maybe different therapeutic considerations, like for instance for TEK and angiopoietin 1, if they don't have a Schlemm's canal or defective Schlemm's canal, maybe a goniotomy isn't the first surgery that should be performed. So you've got this wealth of sort of patients and a database of that, and we need to just bring it forward and actually start doing genetic studies in a very thoughtful and standardized way, in my opinion.
Oh, it's a great idea, and hopefully we'll be able to follow through with some of that. Somewhat related to the group is another question here with an example referring to the Muscular Dystrophy Association, which is actually also a group which is somehow analogous to CGRN. And they put in the question here that this association helps with the cost of genetic testing. And, I might direct this to Peter, actually. Peter, do you want to comment on CGRN and cost of testing?
I think the broader question you want answered is, should we be doing any kind of advocacy work? And the two things I can say is yes, and I don't know how. So I think we should have a larger conversation with the rest of the network. And I think there are people who are part of this endeavor who have the experience and the charisma to lead the kind of effort that can provide services to these young patients, and similar to the muscular dystrophy example. And so, we are always looking to see how else we can grow in our organization, in our little pediatric glaucoma center. And this question kind of hit the nail on its head. Advocacy is perhaps the logical next step, in addition to research, teaching, and clinical care.
Absolutely. Okay. So I think that let me just make sure there isn't any other pressing questions. Oh, is there any hope for gene therapy for congenital glaucoma in the near future?
Absolutely. I mean, I think it's, as I mentioned, I think that gene therapy directed at myocilin is sort of the lowest hanging fruit. There's a very clear path to, a gene editing approach for myocilin. I think that there's also a very clear path for gene-based kinds of therapies for, for TAC and angiopoietin 1, as they target a very specific pathway. Even CYP1B1 should be a relatively straightforward kind of gene replacement issue. I think there are just some, potentially complications there just because of, expression in various tissues outside of the eye and so forth like that, but not insurmountable. So I think that gene-based therapies have lagged a bit in glaucomas. I have a little broader view, not just congenital, but early onset. But I think that there is, definitely the potential for this approach in the future.
I would agree.
Okay, I think we're getting towards the end here. Peter, if you have any closing comments that you would like to add?
Heartfelt thanks to our wonderful, wonderful speakers, and those of you in the audience don't know, they've also been amazing mentors who are so selfless with their time, knowledge, and resources in helping me and the Balkan Center launch some of the research projects. And again, thanks to the Kolokotrones family. I think this was an absolutely amazing inaugural symposium, many more to come. Pretty soon people won't need to do a pediatric glaucoma fellowship anymore. They just need to subscribe and watch. So with that, thank you all for being here. I think we can now jump over to the coffee chat link for those of you who want to schmooze and socialize more. And until next time.
Thank you. Thank you for organizing, Peter.
No,
and
thank you.
Alana.
Yeah, thank you.
Great
Thank
opportunity.
you, Peter and
Thank you.
Elena. Thank you. Thank you, Janey as well.